Happy Friday. Thank you for joining us for the Spring ATE Project Talk Series 2024. My name is Deanne Krupa, and I am the Program Manager with the Innovate Bio Grant at Austin Community College in Austin, Texas. Innovate Bio is the National Advanced Technological Education Center focused on biotechnology. Both Innovate Bio and this series of talks are funded by the National Science Foundation through the ATE program. We encourage and support ideas, innovations you have for educating biotechnicians and encourage you to consider applying for future ATE funding. Please feel free to reach out to us. And as Sandy mentioned, this presentation is being recorded and the video will be posted on the innovatebio.org website by next Monday afternoon. The presentations allow for back-to-back presentations and a short Q&A for both, or actually all three presenters at the end of both talks. And lastly, we will put a couple of links in the chat. The first is a link for additional information about future ATE project talks. And the second is a link for you to complete a survey, which will help us in planning for future events. So it is with great pleasure that I introduce our first ATE talk speakers, Dr. Joseph. Oh, and I'm so sorry, I'm butchering your name, and I practice this. I'll just say Dr. Joseph and Dr. Casal, so I do not butcher their names, and I apologize for that. Their presentation titled Final Outcomes of Dual Credit High School Community College DNA Sequencing and Genomics Project. The first part of the talk will review the final outcomes of the Dual Credit High School Community College DNA Sequencing and Genomics Project, which is providing research-based, hands-on, and real-world workforce experiences for two-year community college and dual credit high school biotechnology students by providing training on industry-level DNA sequencing technology. And the second part, they will give an overview of internationalizing biotechnology education and examples from Japan, Spain, and Ireland. Welcome, Joseph and Casal. I think Casal should be joining us soon.
Excellent. Well, thank you very much for the introduction, Dion, and thank you to Innovate Bio for hosting us today. I'm going to go ahead and share my computer screen so that I can go through our slides. And if somebody can just give me the thumbs up that you can see my computer. Excellent. Then I'll go and begin. Like Dion mentioned, my name is Joseph Olenichek. I'm here in Austin, Texas. I am a dual credit biotechnology instructor. So I'm on a high school campus called the Liberal Arts and Science Academy. And I'm also an adjunct professor at Austin Community College. So my students get dual credit for the courses they take with me for both high school as well as college. My co-PI on this project is Kasao Chidre. He is a professor in the biotechnology department at Austin Community College. Like Dion mentioned, the title of our NSF ATE project is Dual Credit High School Sequencing and Genomics Project. And the first part of our talk, I'm going to focus on sort of the outcomes of this three-year project, since we are in year 3 and we're actually about to finish this project next month in April. And then the second part of the talk, I wanted to talk about opportunities or examples of how you can internationalize your biotechnology education program through some of the programs that I've been involved with over the last few years. So without any further ado, our biotechnology program at ACC has a similar overarching mission to all the biotechnology programs nationwide. Really, we're geared towards meeting the needs of the local workforce, and we do that by recruiting students at the high school level as well as at the two-year level to complete degrees and certificates, and then placing them in workforce. ACC specifically offers 3 certificates and degrees. First is the level 1 certificate, which is sort of a fast track certificate. And this is the certificate that I offer to my dual credit students on the high school campus. They also have a traditional 2-year applied associate's degree in biotechnology. And then lastly, they offer an advanced technical certificate, which is a post-baccalaureate certificate intended for students who have a 4-year degree, but want to come back and get retrained to become a biotechnician. So our NSF ATE project started 3 years ago, and all 4 of our goals for our project really tied in well to the overarching mission of ACC. In other words, we wanted to support that mission of getting more students into the program, getting them to stay in the program, complete the program, and then move to workforce afterwards. Specifically for our project, our 4 goals were the following. One was to establish a DNA sequencing and genomics facility on a dual credit campus. We accomplished that in year 1 by purchasing instrumentation from Applied Biosystems. Specifically, we have a Seq Studio genetic analyzer, which is DNA instrumentation that's used to do automated Sanger sequencing. We also purchased consumables and other supplies that we needed to run that sequencing facility. The best way to think about our sequencing facility on the high school campus is sort of a mini core or micro core, if you will. It provides sequencing services that you could get at a core facility at, say, a large university or a large research institution, but really just supports our students' coursework. Students from my dual credit program, as well as students from the 2-year community college program, use that sequencing center in years 1 and years 2 of this project. And then in year 3, we got supplemental funding, and we actually expanded our capabilities and put a second sequencing facility in at the Austin Community College Eastview campus. So this year in year 3, 2-year students worked at that facility, and my high school students continue to work at the facility we established in year 1. Our second goal for this project was to develop course curricula, focusing on student-centered DNA sequencing technologies. And for that, we collaborated with Cold Spring Harbor DNA Learning Center. They have a very robust program for DNA barcoding, and we worked with them to modify a lot of existing protocols and workflows that they had to make them specific to our lab, and also to put, in addition to that, instrumentation competencies on DNA sequencing equipment, as well as some other student research-led components to those modules that we worked with our students. So we started that in year 1, iterated in year 2, and we finalized those in year 3. We have shared those with several other colleges as well as several other high schools. But if anyone on this talk is interested, I can share those materials with you as well. Third, we wanted to increase the recruitment, retention, and certificate completion for all students, and especially for traditionally underrepresented minority students. And overall, we were very successful in that task. We had 373 students total participate in the project over all 3 years. And I'd like to highlight that from years 1 to year 3, we did have a 25% increase in underrepresented minority students participating in our program. Lastly, we wanted to disseminate our findings through the Innovate Bio community, which is what we're doing today. And like I mentioned, I'm very happy to share anything that I share in today's presentation if you are interested. Kasao, are you on the call? Did Kasao join us? All right, so Kasao, my collaborator, co-PI, unfortunately could not make it. He was supposed to do these slides, but I will go through them right now. So just to kind of highlight sort of the key impacts over the 3 years, you'll notice from project year 1 all the way through project year 3, we had an increase in the number of students who were completing that fast track level 1 certificate. Likewise, we embedded all of the curricula that we developed for DNA sequencing projects within Biology 1414 and 1415 in dual credit, as well as 2-year courses. And we had a steady increase in the number of students at the high school level, as well as at the 2-year program over the 3 years of the project. Again, I mentioned it earlier, between the high school program and the 2-year program, we had 373 students who participated, and we had a little over 100 students who completed that level 1 certificate. Another thing I'd like to mention is sort of the impact that we had on underrepresented minority students throughout all 3 years. Over here in the top left is data from year 1, bottom left is year 2, and over here on the top right is year 3. And you can notice that over all 3 years, we had very high engagement with underrepresented minority students. And we did have an increase from the start of the project to the end of the project of a little over 25%. Part of the way we did this on my high school campus, I went out to clubs, specifically clubs that had students who are underrepresented minorities, and I sort of plugged our dual credit program. Our co-PI, Casal, reached out to local universities, specifically Hudson-Tillotson, which is a traditionally Black university here in Austin, to recruit students into the 2-year program, which is how we sort of increase this engagement over the 3 years. To measure sort of student impact of the curriculum that we develop, as well as them learning instrumentation competencies associated with DNA sequencing technology, we use surveys. So we have pre-survey results over here on the right and post-survey results for year 3. And essentially, we had students rank themselves on how well they understood how DNA sequencing works, how well they understood how to use instrumentation and protocols, and how well they understood interpreting DNA sequence data. And you can notice from the start, before they engage with the project materials to the end, there was a significant increase in the number of students who are reporting themselves as proficient, advanced, or even expert in these categories. Next, we really wanted to make sure that we retained students in the program and at a minimum got them to complete that level 1 certificate I mentioned earlier. After they went through the DNA barcoding module developed by this project, we saw a huge increase in the number of students who were interested in continuing in the program and completing that level 1, which is part of the reason we were so successful in having over 100 students over all 3 years complete that level 1 certificate. Finally, all ATE projects are concerned with sustainability, and we are likewise concerned with sustainability. So in the fall 2023 semester, I started a nonprofit called Loss of Biotechnology. I registered it as a nonprofit corporation here in the state of Texas, and then applied to the IRS for 501c3 exemption. And we were granted both of those in the fall semester. And then we reached out to our local philanthropic community here in Austin to raise donations to kind of keep both of these sequencing centers running in perpetuity. And so far, we've raised $30,000. So we are certainly going to continue these efforts over the next couple of years, and we hope to continue to raise more money to keep this project going after the life of this grant in August 2024. Here are just some pictures from our dual credit sequencing center. This is me with my students showing them how to load cartridges into our sequencer. Over here, we have some students who are actually working with the device, as well as some students who are prepping some samples in our lab that will later on go into our sequencer, just to kind of give you a sense of what our space looks like. And that sort of concludes kind of the updates I wanted to give you guys on the overall outcomes for our project over the 3 years. I will go ahead and transition into the second part of my talk, and then after that, we can do some Q&A. So for the second part of my talk, I wanted to talk about examples of ways of internationalizing your biotechnology program. And specifically, I wanted to highlight 3 programs that I've been involved with over the last 3 years as mechanisms to do so. My first example is going to be from Spain, or excuse me, from Japan. And then after that, I'll talk about Spain, and then lastly, Iceland. So one program I encourage you guys to apply for. I just checked the website this morning before this talk. They are still accepting applications, and there are some application deadlines coming up in July, as well as in November, is a program called the Secure Science Exchange Program through the Japanese Science and Technology Office. And essentially, this is a program where they will support student as well as instructor funding to come from the US or from any country to Japan to work with professors and students in Japan. Specifically, this program is targeting technical colleges as well as high schools. So anyone in the ATE community would be eligible to apply for it. And basically, to apply for this program, you reach out to a collaborator in Japan. You work with that collaborator to develop some sort of program for your students and yourself. And then the collaborator in Japan applies to the program on your behalf. We did this with my students who were taking a quality assurance course. My students got really interested in how sort of regulatory affairs differed in Japan versus the US, and more specifically about how pharmaceutical drug trials differ between Japan and the US. So together with a group of 6 of my students, we reached out to Pfizer Japan and started a collaboration with them. We met with some people from the quality assurance department in Pfizer Japan, and they were willing to host us and willing to mentor the project that my students wanted to do. So essentially, my students wanted to take data from clinical trials here in the US for metformin, which is a drug used to treat type 2 diabetes, and see how those clinical trials were predictive of post-clinical trial adverse effects that patients reported using that drug. And they wanted to compare that to what was going on in Japan in terms of their clinical trials and how often people report adverse effects post-clinical trials. And sort of the underlying hypothesis here was that Japan has more of a homogeneous society, where the US is more of a heterogeneous society. And as a result, they expected to see differences between how predictive those clinical trials were. So again, we reached out to Pfizer. We put a week-long program together. And in February of 2023, I took all of these students over to Tokyo, Japan, and we spent a week there working with scientists from Pfizer. So we were at Pfizer's office for 2 days. And then after that, we went to the PMDA, which is the equivalent of the FDA for Japan. And then on the last day of our program, we were at Kitasato University working with graduate students who look at similar data to the type of project we were working on. It was a great experience for my students, especially for a course like quality assurance. It really allowed them to kind of get some hands-on experience and also sort of internationalize their perspective on what biotech looks like from a quality assurance perspective in another country. In addition to that, some of these high school students had never left the state of Texas and never left the country before. So it was a big trip for them and had a huge impact on sort of their worldview. So again, this is something I encourage people to apply for. The application is open now, and if you have other questions, I'm more than happy to share our application with you or give you some more insights into this program. Another program that I was involved with at the international level was funded by a Dear Colleague letter from NSF. So specifically, I got supplemental funding to the project that I just presented on. And the purpose of this Dear Colleague letter was to get technician training programs in the ATE community to go over and visit other programs in different countries and sort of see how our system compares to their system, as well as to bring some of that back to our students here. So I applied for that supplemental funding, and specifically I was in Spain for 2 weeks, and I visited a university as well as some small companies in a science park in Barcelona. And then I visited a university, the National Research Center, and some small companies in Madrid. And during this time, I collected a lot of fieldwork data in terms of interviews, videos, and pictures, and synthesized all of that information into a learning module that I then put into Canvas and implemented with my students in February of 2024. Really, the purpose of that was to broaden their perspective on what biotechnician training looks like in Spain, opportunities associated with that, and sort of what biotech looks like in terms of global interactions between companies that have a presence in the US, have a presence in Spain, or have a presence in Spain only. So that's another program that I encourage people in the ATE community to get involved with, and I'll share that information in the chat afterwards. Lastly, I just recently returned last week from Iceland for this particular program. This was through Fulbright. Myself, as well as my co-researcher, Teresa Kumaraswamy, who is a biotechnology instructor here at Anderson High School in Austin, Texas. We're both former Fulbrighters, so we had done semesters in the Fulbright program in the past. And Fulbright had a follow-on funding opportunity called the Global Teacher Grant. So for this grant, we wrote it to go over to Iceland to look at examples of green and blue biotechnologies that they're developing in order to mitigate the impacts of climate change. We had a fantastic trip. We visited multiple sites for companies that are doing different levels of green and blue biotech. We also went to MATIS, which is the National Research Institute for Biotech right outside of Reykjavik. And again, we put all these materials together into a Canvas learning module for our students to internationalize their perspective on green and blue biotech, as well as sort of to expose them to what biotechnology looks like in Iceland. And with that, I will sort of conclude my talk there.
We're going to hold questions until after the talks, Uwe. So if you want to put it in the chat, but you can do that, but we'll have questions after Christy's talk. And I see we already have one question in chat too.
Thanks, Sandy. Can everybody see my screen?
Perfect.
Great. Okay, so I'm Christy Schuter-McGuire. I'm the Dean of Biomedical Studies at the Wistar Institute and PI of the NSF ATE grant, Expansion, Curriculum Evolution, and Enhancement during Biotechnician Training, or EXCEED BTT. So first, I'm going to go through these first couple of slides quickly because many of you have heard me talk about this project in the past. But the Wistar Institute is our nation's first independent biomedical research institute, named for Dr. Caspar Wistar, who was a physician, anatomist, and teacher at the University of Pennsylvania. And his great-nephew, Isaac Jones Wistar, founded the Wistar Institute to house his great-uncle's collection of anatomical models and to initiate any other work for the increase of original scientific knowledge to do biomedical research. So at the end, I can put this link in the chat, but we have a great history of science and scientific discoveries having been made at the Wistar Institute, including development of the first standardized laboratory animal model, the Wistar rat, as well as vaccines for rubella, rabies, and rotavirus. But we are perhaps best well known for our basic cancer research. So the Wistar Institute was named a National Cancer Institute, or NCI designated cancer center back in 1972 and has continuously held the designation. So here's a picture of the Wistar Institute. We have our original 1892 building, completed in 1894. And then when we became a National Cancer Institute, we put on the Cancer Center Annex. So we now call the original building our East Building, the Cancer Center addition our West Building. And in 2014, we put on our most modern laboratory space in our Robert and Penny Fox Research Tower. Those are 3 interconnected buildings that are all on the campus of the University of Pennsylvania, but Wistar is independent from Penn. So I oversee our Hubert J.P. Schumacher Education and Training Center, where we have programs for high school students, undergraduate students, graduate students, and postdoctoral fellows. Today, I'm mainly going to focus on programs for undergraduate students, specifically our biomedical technician training program. And Wistar started this biomedical technician training program back in 2000. So Dr. Bill Werner, who is Professor Emeritus and now retired Director of Outreach Education and Technology Training, started this program with Community College of Philadelphia back in 2000. And it was originally a 2-summer program. So Community College of Philadelphia students came to the Wistar Institute for 7 Fridays. And then spent 3.5 weeks in 2 lab experiences, and then came back the following summer for a 12-week lab internship. And we had some really great outcomes. So a 77% completion rate, where 46% of those who completed our program were starting related employment within a year, and 64% continued their education. And we also had some great qualitative stories. So I actually was a faculty member at Community College of Philadelphia who was on the partner side of this program, taught Kwasi Gyampo at Community College of Philadelphia, recruited him into the biomedical technician training program. He stayed working in an HIV research lab at Wistar, finished his bachelor's degree from Temple University, worked at Wistar full-time for a few years, and is now pursuing a PharmD at the University of Pittsburgh. So I came over to the Wistar Institute in fall 2019, and the first summer that I was supposed to lead the program was 2020. And due to the COVID-19 pandemic, we couldn't start a new cohort, but I had spent the time recruiting and selecting students into the program, and I still wanted to find a way for those students to complete the program on their original timeline in the summer of 2021. The pandemic also allowed me to do a deep dive into our data and see that even the 23% of students who didn't complete the program were often success stories because they had already found jobs with just 1 summer of lab experience, or they transferred to a 4-year college or university where they didn't have another summer to return. And we had always had students who accelerated the program completing an early internship either the fall or spring after that first summer. And those students actually had some of the best outcomes in terms of employment. So I applied for and received one of the inaugural grants from GSK's Philadelphia STEM Equity Collective, a 10-year initiative to increase the number of Black, Latinx, and women Philadelphians entering STEM careers. And we piloted an accelerated BTT program in summer of 2021. I wish I could say I waited for the outcomes of that, but you all know how grant funding works. And so we had already put in a proposal to the NSF for this ATE expansion, curriculum evolution, and enhancement during biotechnician training before we had the results. But thankfully, that one summer program turned out to be a good model. So in terms of the goals for this program, for program expansion, as I said, we had worked with Community College of Philadelphia since 2000 with a cohort of 12 students. And what we proposed to do over the 3 years of this ATE project was to increase the number of community colleges that we worked with and also the number of students we were serving in our program. So in 2022, we added Montgomery County Community College. In 2023, we added students from Bucks County Community College and Camden County College in New Jersey. And this summer, we are adding students from Delaware County Community College. So this will give us a nice regional reach for our program, and it really is indicative of where the biotechnology workforce comes from in Philadelphia. The second goal was to evolve the curriculum, and that was to teach the relevant laboratory skills in the context of authentic research actually happening at the Wistar Institute. So my colleague Maureen Murphy at Wistar looks at variants of the tumor suppressor P53. And rather than looking at why these variants increase cancer risk, which they do, Maureen really focuses her research on finding new therapies to more effectively treat individuals with certain hypomorphs or variants of the tumor suppressor P53. So what we do is we work with cell lines that have the African-centric variant, S47, and we follow up on lead compounds that have been identified in a drug screen to, again, more effectively treat the variant cancer cells better than the wild-type cells with the more common form of P53. And through that project, we're taking students through pipetting, reagent preparations, DNA extraction, PCR, molecular cloning, RNA isolation and RT-qPCR, protein purification and quantification, gel electrophoresis of both nucleic acids and proteins. The protein gel electrophoresis is followed by Western blotting and detection, a enzyme-linked immunosorbent assay, or ELISA, bacterial culture associated with the molecular cloning project, and a number of mammalian cell culture techniques and assays. That takes place in a laboratory orientation during the first 2 weeks of our program in our training lab at the Wistar Institute. So we have 2 bays here that have stations set up for 20 students. We also have a place where we can gather students around to talk about the theory behind the laboratory techniques and more about the project that we're working on in this area of the lab. And then right across the hall, we actually just moved from this original tissue culture space where we had 3 biosafety cabinets or laminar flow hoods to teach cell culture to a new room with 4, to help as we expanded. And then the third goal was to enhance the program. And again, we made a number of enhancements. So we did keep that accelerated model of a 1 summer program. Again, starts with a 2-week laboratory orientation where we are teaching that skills-based curriculum on with authentic Wistar research projects. But that 2-week lab orientation is followed by 2 5-week lab experiences. And we really now make sure that each student has one academic research experience and one industry or core facility experience. We also enhanced our program with online resources, including an application system, a learning management system. We use Canvas now, and an alumni networking system utilizing Toucan Tech. So here are our 2 NSF-supported cohorts, our 2022 cohort and our 2023 cohort. So the certificate comes from the Wistar Institute, but our program is also a state registered pre-apprenticeship, so students receive that credential as well. So looking at our outcomes in gray here, I have our historical outcomes. So the data from the cohorts 2000 to 2019, or 160 students. The 12 students that participated in the accelerated pilot program in summer of 2021, and then our 2 NSF ATE supported cohorts in 2022 and 2023. And I think the thing I am most proud of is that we have had a 100% completion rate over the last 3 summers with the program in the 1 summer format. Although we had an initial drop in the percentage of students who were both participating and completing who were underrepresented, that seems to be coming back a little bit post-COVID, and the same with the percent of our participants and completers who are women. We are also meeting or exceeding our historical outcomes in terms of the number of students starting related positions within a year of program completion, and continuing their education at either the community college or a transfer institution. We also ask the mentors of the students in the lab experiences to assess the competencies, and our students are averaging between a 4 and a 5 on a 5-point scale for competencies like following and practicing safe laboratory procedures, using reagents and lab equipment safely and appropriately, following and understanding protocols or SOPs, calculating and preparing solutions correctly, planning and setting up experiments properly, using technical skills and with sterile technique and practices, collecting and recording data accurately, maintaining a laboratory notebook, and now we've also brought in electronic notebooks for this coming summer. Begins to analyze data and troubleshoot, communicates results to a supervisor, and demonstrates professionalism in interactions with colleagues. We also use a validated survey, student assessment of their learning gains, undergraduate research student self-assessment, or the SALG-URSA, and have found that students have made significant gains in terms of thinking and working like a scientist, personal gains related to research work, gains in skills after an initial drop with the first 1 summer version of the program, and gains in attitudes and behaviors as a researcher. And we're moving more students to agree or strongly agree that the program and doing research in the program confirmed their interest in their field of study, clarified the field of study they wanted to pursue, prepared them for advanced coursework or graduate school. And then here in purple, really importantly for us, that the experience is preparing students for a job. And so I have another great qualitative story. One of our students from the 2022 cohort, Tyler Driscoll, was featured on Good Day Philadelphia. I think it might, oh, see, I knew this would happen. I'm going to unshare and reshare my screen to be able to show this to you.
Thank you, Christy, for sharing your ATE project talks. So at this time, we have about 20 minutes, and this is your opportunity to ask questions. But I do know that we do have a few questions that are in the chat. And I know that, Joseph, you did put links, but I know that Laurie, you asked a question regarding certificates.
Yeah, the question was just if level 1 meant anything. Just
Okay,
I'm not familiar.
yeah, so level 1 is just a fast track certificate. So it's 18 hours of coursework. They take 2 introductory courses, a quality assurance course, and then the capstone research internship course. So it's a local term that we use for ACC. It's just called a level 1 certificate in biotech. I did put a link in there to the coursework that they have to take. But to answer your question more directly, there is no level 2. It's just the level 1, and then they offer the 2-year associates and then the advanced technical certificate. Thanks for the question, though.
And then there was another question from you about providing examples for green, blue, and blue technologies.
Yeah, and I put those in the chat. In short, green biotechnologies are agricultural or plant-based biotechnologies. Blue biotechnologies are aquatic or marine biotechnologies. In the context of Iceland, they're doing a lot with bioprospecting from marine biotechnologies, as well as from hydrothermal vents. So you can imagine there, it's a very geologically active country. So we visited several geothermal hot springs. And when we were talking to researchers at Mattis, a lot of them will go out to those hot springs and sample for algae, bacteria, other microorganisms to try to look for novel enzymes that are thermostable. The other interesting component to us, myself as well as Teresa as researchers, is when we visited their bioprospecting lab, they had incubators that were set at 100 degrees Celsius. They had a whole prep system for antibiotic bacteria. There's really a lot of stuff that you don't normally see in a lot of research facilities. So that was interesting to us. In terms of green biotech, Iceland in particular is very, lucky to have mostly all of its power produced by geothermal or hydrothermal. So they're more or less carbon neutral. And as a result, they run these gigantic greenhouses to produce most of the food for the country. And a lot of those are carbon neutral as a result of cheap electricity. So we visited some of those sites. And there's even some biotech companies who are growing genetically modified crops in those greenhouses that you find all throughout Iceland.
Thank you. And Shigar, you had your hand up.
Yes. Hi, Joseph, and who was the other speaker? Christy. Both had wonderful presentations, and I commend how much hard work you put into arranging all of this. I am Senior Director for the Shared Research Resources at Mount Sinai Hospital. I oversee operations for a lot of core facilities here, and I'm trying to have a collaboration with the community colleges in New York City, where the students would come and do projects with our core facilities. The idea is to develop a biotech workforce in the New York City area. So I just want to understand how much time do the students spend in these core facilities or labs? Is it only during the summers or during the school year? Do they get any stipend, and what are the sources of the stipends?
Yeah, thanks for the questions. So during our program, because of the NSF funding, we do have funds to pay the students. And the current rate at Wistar is $14 an hour. So students are enrolled in the program. They're being paid by Wistar $14 an hour. And then their community colleges are also giving them credits for the experience. After that 2-week laboratory orientation, they're doing 2 5-week lab experiences. So, some of our students are getting to spend 5 weeks, and it's, full-time 9 to 5, Monday through Friday, in either the industry lab or core lab, and then also an academic research lab. because of those lab experiences, we actually coach the students to treat that as a 5-week interview. And so many of the labs keep the students on, part-time if they're still continuing their education, or have been willing to hire them full-time. And I think, Sandy knows this story. It's one of my favorite things. Our entire genomics facility at the Wistar Institute is staffed by graduates of our biomedical technician training program. And we also have, other core facilities both at Wistar and, at our cores at the University of Pennsylvania and Children's Hospital of Philadelphia Research Institute. So, core positions have been really great fits for graduates of our program from community colleges. So I'm happy to talk to you more about that. And I've also talked to some other people in New York, so maybe we can get you all connected.
Yes, yeah, that would definitely be good. And I have your name, and yeah, I'll find your email address and
Yep,
get
I can
back
put
to
it right in the chat.
you. All right, thank you.
Shakar, are you part of the ABRF?
Yes.
So Todd Smith,
Mhm.
collaborator on our grant,
Oh,
he
okay.
will be at the
So,
ABRF meeting in Minneapolis.
oh, yeah, I
And
will
he
be
is
there.
working
Yeah.
on a consortium to connect Innovate Bio and the core facilities.
Okay, so yeah, I am aware of his work, the grant, the involvement of Sheena Miche and all that.
Yeah.
Yeah, so, but this is like hearing lifetime, the real life experiences. So this is, yeah, wonderful.
Yeah. Anyway,
Yeah.
just make sure to reach out to him there, because I think
Yes,
I
yes,
don't know.
definitely.
Yeah, I'm not sure how if there are community college people presenting, but he often tries to organize that too.
Yes, last year he had organized for Massachusetts area,
Yeah.
the community colleges, but in New York, we don't have that robust a program to present something like that. So yeah, I'm not sure if something from this area is there.
Yeah.
Okay,
Well,
yeah.
we're glad you could join us.
Yes, yeah. And Christy, so this 5-week program, it's 9 to 5 during the summer.
Yeah, so the whole program is 12 weeks.
Okay.
Although actually this summer it's a 12-week program spread out over 13 weeks because we lose so many days with the summer holidays. But each of those lab experiences is 5 weeks, in general, 35 to 40 hours a week in the lab.
Thank you. And same question for you, Joseph. How much time do your students spend in the core facilities? What's the program like?
Yep. So it varies a little bit depending on what course a student is taking.
Mhm.
At HCC, part of that level 1 certificate, there are 2 introductory courses, Biology 1414 and 1415. So the curriculum that we've developed is around DNA barcoding, and specifically a 3-week wet lab module that we embed within those 2 courses. So for example, students who are just getting initially trained on how to use sequencing instrumentation and the associated sort of equipment and reagents with it, they get sort of a brief exposure to that in those 2 courses. One of the follow-on courses that we offer is a research internship in biotechnology, 2486. And for that, we have students who do semester-long program or projects using the core facility. So for that, they're actually attending a course twice a week for 2 hours, and they're working with the sequencer there. I have had a few students who from my program have moved from our sort of training program to the University of Texas at Austin, has a large sequencing core there, and a few students have worked in that facility as leaving from our program. So that's sort of what it looks like there. Kasal, you can probably talk to that a little bit more about how you guys are using the sequencer over at ECU campus as well with your 2-year students.
Yes, yeah, with our 2-year students at ACC, usually it's a semester-long program. And then throughout the course, for example, 1414 or 1415, so they have to take a lecture. And then in addition to that, there's labs. And throughout the semester, they have to, they get a chance to practice using the sequencer. And then they meet twice a week, 3 hours a day.
Mhm.
And then this allowed them to really run all the whole project. And then the project is divided in modules, so which allow them to run things in step by step.
Okay, thank you. Yeah, it's extremely useful, all this information.
Mhm.
Thank you very much for the questions. Nice to meet you.
Before I miss the chance, I do want to ask everybody to please complete our survey. We have, it really helps us with planning, and yeah, I mean, not every ATPI is as spectacular as Christy and Kasau and Joseph, but we generally have some really good talks. And oh, I also want to be sure to congratulate Christy on the new NSF ATE grant that was recently funded. Okay,
Good.
Uwe.
Christina,
Yeah, briefly, Joseph, how
Congratulations,
did
Christy.
you fund how did you get the funds to establish a sequencing facility at your location?
That was through the NSF ATE project that we talked about. So we had funding in there for the instrumentation, as well as money for consumables for those first 3 years. Now we have an instrument and facility at the high school campus as well as at the Eastview campus. And moving forward, we're going to use our nonprofit to purchase the consumables to keep those two facilities running.
Yeah, smart move. Thank you.
Mhm.
Thank you.
More questions. So impressive.
And thanks. And Sandy mentioned it. I will say so. You know, we did just receive a second ATE grant that will help us continue the expansion of our biotechnician training program. We're going to we called this one Tri-State Exceed BTT because of the interest and the expansion in New Jersey of the biotechnology industry. We're going to add 2 more community colleges in New Jersey and then Del Tech. Again, it's really, I think, more capturing, you know, our regional life science workforce, you know, where we, you know, have people crossing Pennsylvania, New Jersey, and Delaware state lines all the time.
No, congratulations. That's wonderful news.
Yes.
And next week, we will have talks from David Fernandez at Northern Virginia Community College talking about their project. And I will be talking about this summer's antibody engineering hackathon and about the exciting projects that we're going to be doing in August. More questions?
thank you, Sandy. And thanks again to all of our Project Talk speakers today. It's always great to learn about what you are doing, the wonderful opportunities that you're providing our community college students and high school students that are available through your projects. So as you can see, the chat is filled with a lot of different links that we will stay on until 1 o'clock in the event that you have not had a chance to copy those links. But we will also have the recording available on the innovatebio.org website that Sandy mentioned earlier. And again, we respectfully ask that you complete the survey. Again, another link posted in the chat. That will help us in planning future project talks and events through Innovate Bio. I thank you again, and as long as there aren't any additional questions, thank you again for participating, and we again encourage you to register for future project talks. Joseph, Christy, and Kasau, thank you. We appreciate your time and your expertise. And happy Friday to everyone, and best wishes for a wonderful Easter.
Good weekend, everyone. Thank you.
Thank you.
Nice meeting you.
Have a great weekend. Thanks, everyone.
Thanks.
So.